just it's a lot of stuff. But this was another contributing factor. The okra protein is called opacity. I'm not gonna make you read that, but it's actually called, we just call it okra protein. So the okra protein allows for attachment to the whole cells. But what's crazy about this particular protein is that it's gonna actually allow the bacteria, because it's found on the membrane, the outer membrane of the protein, and so it's going to allow it to be antigenically diverse. So the antigens that normally would have, they can change it up. Right? They can be a little bit different from what they're supposed to be, which makes it hard to treat it, right? If I thought I knew what the antigens were, and I have an antibiotic to target that one, what's the type of antibodies to target that one, they don't really pair up if you change the antigens, right? If you change the target, then how am I going to target it? Alright? So the okra protein allows for the gonorrhea to persist in the female genital tract. That's why with gonorrhea, a woman can have it, sometimes not know it, because she doesn't display symptoms for that particular one, but the particular bacterial infection can persist. And the issue with that one is that for gonorrhea, chlamydia, and many other STIs, if you have it, get it treated, because there's this thing called pelvic inflammatory disease. You don't want that. Alright, that's really worse. Alright? So that's why I tell people, whenever you're sick, get it treated. I know a lot of times we talk about the cost of things, but it's like, I kinda want you to live too though, so you gotta figure it out, you know, and then medical bills, no, I'm not gonna tell you about that. I'm gonna tell you something, it's true, the true statement, but I shouldn't tell you. Alright, so with that being said, I think I deleted that, hold up, let me erase this. I thought I erased it, I guess not. Ooh, I wanna keep that, that's fun, I wanna keep that. I thought I went through there and erased some of the stuff, not all of it. Okay, we'll keep some of it, some of it's fun. Yeah, I think I wanted to keep the drawings because they're fun. Yeah, they're fun. Okay, so we're looking at this slide right here. We have let's finish up with the waxy leaf. I forgot about Mycobacterium tuberculosis. So Mycobacterium tuberculosis has this substance in its cell wall called mycelic acid. We talked about this in the lab, acid fast stains, right? So the acid fast stain, the reason why you have to do that is because they have this mycelic acid in its cell wall. It's waxy, doesn't really want to absorb those stains with the gram stain protocol very well. So that's why we use the carbofusion, the methylene blue, and the acid alcohol for these guys, right? For an acid fast stain. And with the waxy mycelic acid, it allows the bacteria, Mycobacterium tuberculosis, to resist digestion by beta cells. So those white blood cells can't really target it as well as it like to, courtesy of the waxy mycelic acid. Alright, so it's a way for us to identify the bacteria, but it's also a way for the bacteria to be protected, right? So it's like, oh, it's like great for the bacteria in both ways, right? So it's like you can't find me, and then you can't kill me. It's like, it's great. So the bacterium is great, not so much for us as a host, right? So enzymes, what are enzymes? Speed up chemical reactions. Are they changing the process? No, they're not changing the process, right? And that's because they're able to be free and don't want to do more chemical reactions over and over and over, right? So with these enzymes, I want you guys to know this, right? So the name's going to tell you what's going on. So whenever we see the ASE, we're going to see the fact that they're going to be an enzyme. So coagulases will be involved in coagulation. So it's going to be involved in coagulating the fibrogenic form of fibrin. So we're going to look at this process in a little bit, though it'll all make sense. So right now, I'm not saying like memorize the slide, the process is going to come together in a couple slides. Alright? So kinases will break down or digest the fibrin process. The process one will break down the kinases. Hyaluronidase is going to be able to break down hyaluronidase. But when talking about the flu shot, the reason why you have it yearly is because you don't know which variety is gonna be present within the population, right? There can be a different strain for that year. And so every year, they actually like go through and they start to collect samples from people that are sick. When we start looking at that around like summer to about like early fall, you start to see which strains are the most popular, the most prevalent in the population, and that's what goes with the vaccine. So that's why a person can take the flu shot and not be affected, it not work because that might be a variety that's not in the vaccine, right? So that's why when looking at the influenza virus vaccine, it's good to take it for the most part because it does give you a level of protection, which you have to understand too that it's not going to be 100% because you might have a strain that's not covered in that particular influenza shot. Right? And so I always tell people that, like, I personally just started taking I only just started taking the vaccine when I was like, I thought I was gonna die. It was like 2019. I had the flu, and I thought that was it. Like I was in the room, just laid out, just smooth laid out, and I was like, I can't do this again. And I don't even take it every year like I'm supposed to. I take it like every other year when I remember to do it. That's really what it boils down to. But I won't lie to you. I'll be honest with you. Like I'll remember, and I'm like, oh yeah, I should do that. And I'll do it. Or if somebody gets sick, like my daughter, she got the flu, I went and got the vaccine that day. I was like, stay back. Give me like 2 days. But the vaccine, I'm in 100%. Because you can, so, and then there are people that'll be like, I got the flu and I got the vaccine. So let me tell you what happens. You really didn't get the flu from the vaccine. What happens is that, very rare, that's rare. What happens is that when you get the flu shot, you get the shot, and then your body is gonna be like, whoa, what's that? Where did you come from? They're gonna recognize that something hasn't been in your body, right? So as your body recognizes that, they're gonna release antibodies to that site. So the antibodies that are going to that site, those white blood, not antibodies, sorry, the white blood cells are going to that spot. So the white blood cells are going there. They're like, whoa, what is this? Destroy it. And so those white blood cells are gonna start making antibodies against the pathogen, right? Which is the flu virus. And so it's going to make antibodies to target the flu right now. It's also gonna make some memory antibodies. And those memory antibodies are gonna remember. So that way, upon a re-exposure to that virus, your body's gonna be like, wait, I know you. I already got something for you. Right? They're gonna send out the antibodies to target, like they really do. Like it's so cute. They're gonna send out the antibodies to target the virus, and then that's why you don't get the virus after you have the shot, if it's one that's in the vaccine. Right? So I've never gotten the flu shot. I've never gotten the flu after getting the flu shot. Right? But I'm also careful too about doing that. Right?
But also they say if you get the vaccine, you can get sick, it's just not going to be as severe.
Yeah, you're gonna get
to train your immune system to fight off the.
Yeah, you're gonna.
It doesn't necessarily guarantee that you're not gonna get it.
Yeah, you're not gonna get the virus. You're not gonna get the virus itself, but you're gonna get sick because your body's fighting off the response, right? Your body's gonna your body's basically like, hey, let's get out there. What's dead?
The vaccine.
So the vaccine is like dead. It's killed. The vaccine is killed. But when I was younger, there was a flu vaccine that was a live vaccine, and I couldn't take it because it had eggs. It was made with eggs, and I used to be allergic to eggs as a kid. And I would have to sit at the doctor for like 30 minutes. They're like, you take the flu shot, you can stay for 30 minutes observation. No. So that's why I wouldn't take it. I was like, no. Alright, so gonorrhea also can change your antigens too. So that's why it's hard to treat it. That's why if a person has, this has happened. This happens where people can have a gonorrhea infection and the antibiotic doesn't work. Right? There's actually a strain. I don't know if you guys know this, but there's a campaign in Louisiana right now. They have signs like all over. They're like, gonorrhea resistant strain. We might not be that serious. Like it's really like all throughout vaccines where it's a resistant strain of gonorrhea and it doesn't work as effectively with certain antibiotics. It's not good. Why is that gonorrhea so much? Yeah, why is it not? Because there's a lot of gonorrhea out there. So why don't they be? So here's some Bruce Lee can be in this. That's a true statement. So, the digestive enzymes speed up the process for breaking down different things inside of the cell that need to be broken down. Alright? So, this lysosome feeds with the phagosome because it wants to break down this bacterium, right? Right? So, the problem though is that some of the bacteria can escape from the phagosome before it actually fuses with the lysosome, or this particular bacterium can prevent fusion of the lysosome with the phagosome. So this doesn't happen. This step of the fusion does not happen because the bacterium can actually just stay inside the phagosome, hang out, and prevent its own destruction. Right? So that's pretty cool. It's like, it's cool, like, in conversation, but it's not good for the human host. Alright, it's not good at all for human host. So salmonella is a good example. So salmonella, we know, is a foodborne illness, right? So that's the one that you get from uncooked or raw foods. So that we talk about the chicken, uncooked chicken, that's not a good thing. So salmonella entering intestinal epithelial cells, intestinal skin cells as a result of rupturing. So the rupturing here, you see of the host plasma membrane will allow for salmonella typhimurium to be taken up into that particular cell. When it's taken up into that particular cell, it's gonna cause infection. Alright, it's gonna be causing an infection. So salmonella is in fact a bacteria that has flagella all over its surface. It's gonna be an enteric we're gonna talk about in a little while. Alright, it's one of those bacteria that has a lot of interesting features. And so when talking about this video, I'll let you guys watch that one later. But we know about biofilms, right? We know about biofilms. We know that biofilms form layers. We know that they're gonna be able to resist antibiotics and disinfectants, right? That's a problem. And they're involved in about 65% of all infections, right? 65% of all infections. So it's like, okay, that's not good. So, what are they able to do? So how do they actually penetrate the host cells defenses? Well, they're able to evade, again, phagocytosis of the phagocytes, right? Those white blood cells. So they play a role in evading those white blood cells. Now, these biofilm bacteria are more resistant to phagocytosis, shielded by the EPS. That's why I put a square around that one, because I'm not gonna be writing out acylated polymeric substance. I'm gonna just simply write out EPS. That's how I am. So the EPS of the biofilm, alright? So the extracellular polymeric substance, the EPS, is gonna be outside of that particular bacteria to protect it, right? So the biofilm provides their protection against the phagocytes, the white blood cells. Alright, so it's great for the bacteria, right? Great for the bacteria. Alright, so things to think about. What do capsules and M proteins have in common? I'm not going to actually about another example, it's just an interesting thing to know about. Alright? And so when looking at this one, I talk about like many vaccines provide risk protection against disease, modern vaccine offer more than 3 months of protection. That's one that we just talked about, right? So why is that? Yep, the antigenic fragments and the variation. Absolutely. Alright? And so these are the ones, we talk about these, how the bacteria can avoid destruction by the phagocytes, the white blood cells, we talk about that. Or Streptococcus pneumoniae, them having a capsule, right? What about Staphylococcus aureus? Let's go back. Oh, you know what? I didn't say specifically what that one particular one was, but that's gonna be for your enzymes right here. So these are gonna be some of the enzymes that's gonna be involved with Staphylococcus aureus. Alright, so Staphylococcus aureus have these enzymes that allow it to avoid being destroyed by the photosynthesis. Alright? And then what about listeria? I'm excited because I saw that. Actin.
escape host defenses. One group of virulence factors are enzymes secreted to help the pathogen to penetrate tissues, allowing them better access to nutrients and helping them to reproduce. Cells that make up body tissues are held together by molecules.
Wait, no.
One way in which pathogenic bacteria
Cause the video cut it a little bit, let me see.
virulence factors
Can you put on, that's the hyaluronic acid right there, I'll just tell you. I'll just tell you.
help the pathogen to penetrate tissues, allowing them better access to nutrients and helping them to reproduce. Cells that make up body tissues are held together by molecules like hyaluronic acid, a polysaccharide that forms a chemical glue. Some pathogens, such as certain Streptococcus species and Clostridium perfringens, secrete an enzyme called hyaluronidase that breaks apart this glue and makes space for the bacterium to penetrate deeper within host tissues. Collagen fibers, found at the base of superficial tissues, are another barrier to pathogens. Pathogens like Streptococcus and Clostridium secrete collagenase, an enzyme that helps them to pass through the barrier by digesting collagen fibers and allowing these pathogens to move deeper into the body. Streptococcus pyogenes, which causes strep throat and necrotizing fasciitis, secretes an enzyme called streptokinase that digests blood clots by breaking down the fibrin proteins that provide the structure of the clot. For this reason, streptokinase is sometimes called fibrinolysin. Some strains of Staphylococcus aureus secrete staphylokinase that do the same thing. Both of these species of bacteria live on the skin. The ability to dissolve blood clots
Alright, so that was that slide, right? So those different enzymes are found in the bacteria on that slide that I showed, those different enzymes. So that's what I want, that's why I want to show this to you. Alright, so I want to show you this one as well with the salmonella and shigella because these are enterics that we're going to talk about. so toxemia is gonna be the presence of toxins in the host blood, alright? And so when looking at intoxication, that means you have toxins within your blood, but there's no microbial growth. Alright, so you have toxins in the blood without the presence of microbial growth. And so we have two overarching categories for toxins. The first one is gonna be exotoxins. Exotoxins are interesting because these are proteins produced and secreted by bacteria. And so these are gonna be those that are gonna be typically released from living cells. Alright, so the bacteria are alive when they're releasing the exotoxins, alright? And so these bacteria are alive when the exotoxins are released, and so these are soluble in bodily fluids, they destroy the whole cells, they can also inhibit metabolic function. Alright, they can inhibit metabolic function, cellular respiration, things of that nature. So they're highly specific for their targets, and some of them are highly lethal too. Alright, so that's what I was saying, like, you're trying to kill the bacteria, okay, cool, but kill the toxin too, destroy the toxin too. Alright? That's why you have to know what bacteria it's gonna be. So, antitoxins are gonna be antibodies against specific exotoxins, and they will provide immunity against that particular toxin. Alright? You'll be protected against that particular toxin. So, toxoids are inactivated exotoxins that are used in vaccines. And so basically, remember I was saying that if you kill the bacteria, you gotta kill the toxins too. So if you're killing the bacteria, you first have the vaccine, right? You have to put something in there to destroy the toxins if it does in fact produce toxins, right? What happens is, if you kill the bacteria and that bacteria releases the toxins, and then see for like a certain time point, you're gonna make that person go to toxic shock. Because you didn't destroy the toxins, right? And that's something else that you have to see. That's why a lot of times giving a broad spectrum antibiotic is not always the best move, right? Because you just give a broad spectrum drug, not knowing what it really is, and then you don't know what it really is, how would you know if that's toxic? Right? That's why I recommend doctors that be like, if I give you that, speak on the business. Stay strong. Stay strong. You know, your doctor, like, stay sick with that. That's really something that you, like, hold up against any stage you have. So, when you look at the exotoxins, these are, again, produced inside bacteria. These are most commonly gram-positive, right? So these are I'll write that down. They're most commonly gram-positive. I'm not gonna say they always are, but they're most commonly gram-positive bacteria. Right? I'll write that down somewhere. And so these are gonna be secreted into the surrounding medium during long phase. So we haven't talked about that yet. So long phase is when there's gonna be exponential cell growth, cell division. Alright? So so log is exponential cell division, and in this particular process, we did talk about log. And yeah, I'm like, wait a minute, I'm thinking like, microbial growth chapter, we did. We talked about the lag phase, the log phase, stationary phase, the stage of decline. We did. That was good. Alright, so during log phase, it's gonna be really, really rapid cell division, and this is when this guy's gonna release where cells are black. Alright? So you have a bacterial cell. This little lovely cell wall, yellow plasma membrane, this cytoplasm on the inside, but it just so happens to have exotoxins. So if exotoxins are released to the outside of the cell still alive. Right? So the cell still alive has to release exotoxins. What does that? Botulinum, this one cell can be made, the bacterial cell can be made those choke points, or not so much the bacterial cell, the whole cell can be made those choke points. It's going to potentially become cancerous cells and then continue to divide and not follow the rules of regular cell division. Alright, so may lead to cancer, not always going to lead to cancer, right? It's a possibility if it damages the DNA. So let's look at these steps for the AD toxin. So the video kind of did a really good job, I think, of talking about it to detail overview what happens with the AD toxin. So we know that DNA is going to facilitate the production of proteins. We know that, right? Because DNA is RNA and protein. So here we see DNA is going to be made, it's going to be making the exotoxin for the DNA, RNA proteins, and so the exotoxin polypeptides, aka proteins, components will come together. When they come together, they're going to be released in that bacterial cell, and when they're released in that bacterial cell, as we saw in the video, they're going to interact with the cell surface receptor of that host, right? The cell surface receptor of that host cell will be in contact with the B portion of the AD toxin. And when it does bind with AD toxin, it's going to be taken up into the cell. So the cell invaginase takes up the AD portion of that toxin. So when it takes it up, we have a little, you saw that endosome, that's basically a body structure that's going to house the molecules, the AD toxin. And so it's going to take it up. This process, I don't know if you remember, but it's called receptor-mediated endocytosis because it interacted with a receptor that mediated the process to take it up, right? Endocytosis, take it up into the cell. And so basically you're going to see the structure being taken up. It's going to migrate to where it needs to go because the portion of it, the A component of it is going to, the AD toxin will be separated, and the A portion of it is going to be involved with gene host cell function. The B portion of that is going to be able to recycle back so it can release and go along in this process all over again to prevent the production of those proteins. When the rest of the DNA, you don't have proteins. So the whole cell is going to be rendered non-functional at some point because the cell can't sustain itself with its own proteins. Alright, so the AD toxin is able to interfere with that process. Alright, so membrane disrupting toxins, as you saw with those that will go in and actually make pores and it makes a hole in the membrane itself, everything comes out, it makes a hole in the actual phospholipid bilayer, everything's still coming out, it's just that there's different ways to make the cell life, right? So what does that look like? So you have the leukocytins, these are here the chemoathanasic leukocytes, right? White blood cells. Alright, so these are white blood cells, hemolysis are going to be reticulocytes, red blood cells, glycogen protein channels, and then streptolysis are going to be hemolysis. So basically, so when talking about strep, we're going to grow strep on some blood oxalates because strep can break down red blood cells. And the reason why streptococcus can break down red blood cells is because they have these hemolysis, right? They have these enzymes that allow us to break down red blood cells. Alright? So that's what they when we talk about the streptolysis, these are the different toxins that are able to mediate strep. Alright? So strep does some stuff, right? That's why I tell people like strep throat is a doozy. You gotta watch that one. Alright, so super antigens, as you mentioned before, so this was really interesting because with the super antigens, as you mentioned, it does kind of trigger off a systemic response because we have these things called cytokines. So we haven't talked about cytokines yet, but cytokines are going to be involved in the inflammatory immune response because super antigens, they're going to cause an intense immune response when those cytokines are released from those cells. T cells. We're going to talk about the adaptive immune response, and T cells are really important for targeting certain infections. Alright? And so with this particular release of the cytokines from T cells, if you have a regular amount of them, they're doing their job as they're supposed to, then yes, it's going to target the invading pathogens, right? The ability. But sometimes people use an excessive amount, those cells produce an excessive amount of cytokines, and that's going to like overextend. So you'll then see things such as fever, nausea, vomiting, diarrhea, shock, and in some cases, not a lot of cases, but in some cases, it's there. Alright, so we're talking about cytokine storms in the chapter 17. cytokines. And so if you have a large quantity of cytokines, that could trigger a cytokine storm. You don't want that because that can cause other issues, right? Nausea, fever, chills, weakness, generalized aches and pains. So it can also cause disseminated intravascular coagulation. Lots of big words. Only just to tell you there's gonna be uncontrolled blood clotting. Alright, so that's basically uncontrolled blood clotting. If you have uncontrolled blood clotting, what do you think happens? You can bleed out. That can potentially trigger a heart attack, but it can lead to severe bleeding. Alright, so uncontrolled blood clotting can lead to severe bleeding. That's why for people that have issues with clotting, we can take medications to help with that process. Yeah, with that particular case, uncontrolled blood clotting, with this particular case. Yeah. All right. So this one can cause endotoxic shock. All right. It can cause endotoxic shock and it can cause life-threatening drops in volume as well as blood pressure. So do you guys know what your blood pressure should be? And it depends too. It's not just the same. Men and women a little bit different. Also ethnicity also can play a role too. That's one thing that I've learned as I've gotten a little bit older. But about 120 over 80, if you drop below 90 over 50, that's too low. Alright, anytime you go over 90, if you go below 90 over 50, that's too low. Alright, so if it drops that low, then that can lead to endotoxic shock. And also, it may weaken, this toxin can weaken the brain blood barrier. Alright, it may weaken the brain blood barrier. So that's another concern when it comes to certain bacteria and endotoxins. Alright, so let's look at that. So what are the mechanisms for the endotoxin specifically? Alright, so we look at this where you have your gram-negative bacterial cell with its outermost membrane, right? And so with its outermost membrane, you're gonna have the LPS layer of it. And so with the LPS layer, it's gonna be part of the outer membrane, the cell off your head. And if the cell is the cell dies, you're gonna create toxin. And the cell wall breaks apart and releases that out into the surrounding environment. So Salmonella typhimurium, Salmonella is the classification of food poisoning. And this one can release toxin. So if someone has that particular infection, then you want to also make sure that they are treated for toxins. Alright? So I saw some of you guys were a little concerned when I said about the disseminated intravascular coagulation. I want to revisit that because y'all look a little concerned right there. So the reason why it's considered a widespread excessive blood clotting in the small blood vessels is because it can actually consume the clotting factors. When it consumes the clotting factors, it can lead to internal and external massive bleeding. Alright, so that's why I want to break that. So with this particular one, disseminated intravascular coagulation. Disseminated intravascular coagulation, this particular one, often fatal or excessive widespread blood clot in small vessels consumes the clotting factors, leading to massive internal and external bleeding for that individual. And so that person, in this particular case, the small blood clots, they develop in capillaries, and so blood flow will reduce as a result, and the clotting factors are going to be basically consumed by the initial clotting. So that way, the clotting process is like small, but when it happens though, it's going to trigger a lot of different factors to be exhausted. So the clotting actually happens, she said in small quantities. Yeah. Look, wrong way. Yeah, small clots. maybe you just don't know. Like, I have to like think about it that way. Just because you're a microbiologist, you just don't know, you know? But ideally though, when it comes to fever, having a fever actually is not always a bad thing. Let's see what happens when you get a fever. Alright? So, when you get a fever, what's gonna happen is that your body's gonna be like, wait a second, there's something going on here, right? So you have a bacteria that's going to be ingested, and so this gram-negative bacteria is gonna be ingested by this particular macrophage. So the macrophage, the white blood cells doing what's supposed to do. White blood cells eat, they eat the bacteria that's gonna be the big pathogen. So great. The bacteria is taken up, the endotoxin also present on the surface of that gram-negative bacterial cell, and so the bacteria is gonna be broken down, it's gonna be graded inside the vacuole, right? It's taken up through this little vessel anyway. And so this little vacuole then is going to release the endotoxins because if the bacteria is broken down, when the cell wall's broken down, the LPS is gonna release, the endotoxin is released, right? So the endotoxin is gonna be released, and then that's gonna cause the cytokines to be released too. So it's gonna cause the macrophage to like produce cytokines, and those cytokines are gonna be involved in the immune response with certain symptoms. So it also release these things. I'm gonna tell you this now. We're not gonna go into the details. Right now, not too many details because very surface level. So interleukin 1, IL-1, and TNF alpha. So TNF alpha is tumor necrosis factor alpha. Those are two other immune responses that's gonna be released because they target certain things in the body. Alright? Target certain things in the body. They're basically gonna be targeting the invading pathogens. So when this happens, those cytokines though are going to be released to the bloodstream. If they're released to the bloodstream, they're going to be transferred from the body, right? The blood goes everywhere. Blood goes throughout the body. And so they're going to travel specifically to the region of the brain called the hypothalamus. What's the hypothalamus? It's the control center of your brain's the thermostat, right? It's the thermostat. It regulates your body temperature to make sure it stay ideally about 37 degrees Celsius. Ideally, right? Ideally. Some people might be a little bit below 98 degrees Fahrenheit. 98, sorry. So some people are a little below that. Some people are a little below right? Nothing too crazy though. You don't normally see people sit at 100 degrees Fahrenheit. That's your normal resting body temperature. That's the problem. Okay? That's the problem. Alright? So in this space though, once these cytokines reach the hypothalamus of the brain, it's going to induce it to produce prostaglandins. So prostaglandins are going to be involved with regulating the body's temperature to a higher temperature to induce that fever. And that's because if you increase the temperature, you can kill off the microbes and pathogens. So that's why I say sometimes a fever isn't really bad. That's your body's way of protecting itself. Alright? I know a lot of times as soon as that temp comes up, y'all straight to Tylenol. Right? And you think your body a chance at all. It's like just give it a little time. But once it gets too high though, you don't even sometimes if it's too much severe pain, don't let it get to you. Don't let it get to you. You'll increase your blood pressure. Don't if you have a severe headache, just watch that it's low, increase your blood pressure. I say for me, my stopping point, at about like 100, if we break 100, yeah, that's too high. Once we break 100, it's 100.1, too high for me. Breaks 100. Right? And my child, I don't even play with my child. Like my child, with her, if she's at my house, we know the thermometer at 99.9 or 99.5, because on a home thermometer, 99.5, it'll be about 99.5. It'll take it down, right? So with my child, I was like aiming temperature, I was like aiming temperature, oral temperature, put up these arms to get a couple of like numbers, because she's a baby, she can't tell. Right? So I just checked temperatures, and for her, I was like under 100. If it breaks 100, we're gonna go take those for Tylenol for 6 hours, go take those bad ones, about to drop her up, get her on. Absolutely. So when talking about the Laos assay, I like this because the limitless aminocyte lysate assay is used to test for endotoxins. Okay, so when looking at this, we have horseshoe crabs. You guys know the horseshoe crabs have two blood? Beautiful. This is the blood of horseshoe crabs. So horseshoe crabs are over on the Atlantic coast, and they typically use them for pollution fields. And so they do so because this blood contains amebocytes, right? The blood in these horseshoe crabs contain amebocytes. Amebocytes are the lice in the presence of endotoxin on the body, right? So this is really, really, really good in the pharmaceutical industry. I put some articles up for you guys. These are also in the PowerPoint for you guys because they were able to make vaccines from horseshoe crab blood. The issue though is that it takes a lot of time and it also exerts a lot of energy for the horseshoe crabs to release a lot of this, to donate, well, not even donate, they're not donating nothing, they have no consent. But for them to,
Really ask for it.
right, no, I did, can we take it? Nobody did that, nobody did that. So, with the hardship crafts, unfortunately, there's a lot of there's some ethical things about it that people talk about. But for the most part though, it's great because it is going to be used for, pharmaceutical companies for quality control checks as well as for making different, drugs. Alright, so it's a really, really cool thing. So, little video on the endotoxins, I'll let you guys watch that one. But I do wanna compare and contrast endotoxins and exotoxins. So, exotoxins, we said, are gram-positive bacteria generally, but also some gram-negative. Remember I said generally, but I didn't say all, right? That's why I was like, alright, so it's generally gram-positive, but it's also gram-negative. That's a good question. You wanna see that? I'll put that on. Alright. You want me to word it like that, but that's a good question. But that sounds like a true false question, right? Like, looking at that though, very clearly, to answer your question very clearly, it's always gonna be exotoxins are found in gram-positive and gram-negative, right? Always. Endo though are gram-negatives. Endo are gram-negatives because it has to be a part of what? Of the gram-negative cell. The cell wall, which is the LPS, and only gram-negative has LPS, right? So that's how you know. Endotoxins only gram-negatives, and it's because of the LPS. Alright, relation to the microorganism, we just said that for the endotoxins, for exo, metabolic product growing cell, basically it's made with a growing cell. For the chemistry, we saw that we had the AB exotoxins, so usually by 2 parts, but we had other ones too though. Right, with the AB exotoxins, we had the superantigens, we also had the membrane disrupting toxins, right? So remember that there are actually more than just the AB exotoxins. And then for endo, we have the lipid portion of the LPS. It's like, oh, that's easy. And so with the pharmacology of these exotoxins and endotoxins, the exotoxins are very specific for a particular cell structure or function in the host, right? So they're targeting certain things, which makes sense, right? They're targeting the cell receptor, then that's gonna have to be very specific. It has to know the cell receptor. If you're targeting the membrane, it needs to know, is it a real, like what kind of membrane is it? Like how do we like destroy that membrane? Very specific, right? Whereas for endotoxins, you're just like releasing LPS everywhere, right? It's not a specific process, very generalized, very broad. Right, very broad and familiar. So they all produce the same effects, unfortunately, with the gram-negs. And then the heat stability, so with the exotoxins, they're not unstable, so we can destroy them, which is good. At about 6 to 8 degrees Celsius. And so we're looking at that, this is the